today. Just people always ask, where is Brigitte working? You know, somewhere in France. So you'll see my address is CEDEX. CEDEX in French means zip code. So I'm sure you all live in zip code, right? In a zip code city, whatever. I've developed all the research I'm going to show you was developed in this institute in Strasbourg, and Strasbourg is a French city which is right in the middle of Europe. So this is Western Europe. You recognize Spain, where Cold Spring Harbor actually will take place next year and was last year. France, England, Germany, and Strasbourg is right here on this black dot. Okay? Next year, I'll be on another continent, as you heard. But all the research was done there and was done in a department that basically where all the teams are trying to understand the genetic basis of pathological situations, basically. And our kind of expertise is using mouse genetics and manipulating the mouse genome in vivo to try to produce mouse models of human diseases and understand the human diseases. So I don't know if you've ever seen this little cartoon, Tintin and the Lotus Bleu. Do you know Tintin? Tintin. This is an opioid dam. The blue lotus, exactly. Opiates actually are.
It's not a kid's story.
No, it's never kids' stories, but every kid used to read them. And I read the stories, all the stories to my kids, and they all know them by heart. So this is the opium den, and the kids realize that opiates actually relieve pain, are incredibly pain relievers, and they relieve all kind of pain, physical pain, you could call it mental pain, psychological pain, social pain, whichever pain you can think about. And you also heard that opiates produce euphoria. So for all these years, we and many other teams have been trying to understand how do they do this and where do they do this. And as you know, our brain is made of millions of neurons. Each neuron connects to 10,000 other neurons, and each neuron expresses at its surface thousands of membrane receptors and channels and sensors to respond to external stimuli and basically to convey the electrical signal. So how and where? And that's what I'm going to talk about. So as I just told you, we basically our approach is to probe this molecular process and now the circuit processes by manipulating the mouse genome and generating these mouse tools, and you'll have data from classical knockouts, a little bit of conditional knockouts, and also from knock-in approaches. So I guess you're pretty familiar with this. I'm not going to go to enter into the methodological details, but basically what I'm going to show you is what you can learn from these animals. So we basically modify the genome, and then we challenge these animals in many behavioral tests and see what we can get from there. So the presentation, we have two parts. So as usually, as traditionally, part one will be more didactic. You'll have a lot of background or type of overview of what opioid receptors do in the brain. First part, clearly, basically to overview the molecular properties of these receptors. A second part on their roles in vivo using knockout mice as tools. And a third part, which will show you a new tool we've been developing to basically see the receptors in vivo. And the second part, which will be a bit shorter, I'll tell you two stories that have not been published yet. Okay, so let's go from there. So, very briefly the history. So, the most active ingredient of opium, of course you know that, is this molecule that was basically discovered in 1805, called morphine. Morphine has strong analgesic and addictive properties, and since the discovery of this molecule, chemists have been trying very hard, so it's 2 centuries now, to develop the ideal analgesic that would not be addictive. this doesn't exist yet. Well, probably will never exist, but it's a little bit more complex than that. But a few years later, 1898, the company Bayer found the molecule. They deacetylated morphine and they marketed the first analgesic that is a non-addictive compound. And you know the name of this molecule? Heroin. So heroin was heroin was put on the market to treat cough, by the way, and was used for any type of medication until medical doctors, took about 20 years, realized that there was a little problem with dependence. So from there, anyway, let's move now to 1973. So this is 40 years ago. And 40 years ago, 3 teams demonstrated that these molecules are able to bind on the brain in a discrete fashion. So you can see that a radiolabeled opioid is able to bind in a reversible and saturable manner. So there should be receptor sites. And there came the concept of a receptor. And since these molecules had receptors in the brain, of course, people ask the question of why would brain would produce receptors for molecules from the poppy seeds, right? We are not born in the middle of poppy fields. Why would we have those receptors? And the search for an endogenous ligand came, and 2 years later, met and luankephalin were isolated, and that were the first opioid peptides. So, from there, it was clear that we have what we call an opioid system, receptors, peptides, and this system clearly modulates pain pathways, hedonic status, reward dysphoria, and responses to stress. And if we would, a simple way to think about the opioid system is to think that this system is in our brain to help us learn what is beneficial for our survival and teach us how to cope with challenging situations. That's maybe the way you could think of this system. So what about the genes, the molecules? The genes encoding the peptide were cloned in the 80s, right, early 80s. And basically there's 3 genes. You know them under the name of POMC, pro-opiomelanocortin. So this gene encodes a large precursor protein which basically gets rise after proteolysis to several biologically active peptides. One of them is beta-endorphin, but there's also ACTH, MSH, and others. Beta-endorphin is the longest opioid peptide. There's another gene that encodes a precursor protein called pre-pro-enkephalin that give rise to several copies of enkephalins. And these are the short peptides. And there's a third gene called pre-pro-dynorphin, encoding dynorphin, which are the basic peptides. And you heard about dynorphins by Charles Shafkin. So we have these 3 genes encoding peptide, and we have 3 genes encoding receptors. So the first gene being cloned was encoded a delta receptor, and this was cloned by the team of Chris and my own team the same day, as you heard, in 92 or 91, no, 92. And from there, homologous genes were cloned. So we have a family of 4 genes that are highly homologous. 3 of them encode opioid receptors that are called mu, delta, and kappa. And the 4th one encodes a related receptor that is not an opioid receptor. It is called the nociceptin receptor, or often in FQ receptor. And I'm not talking about this one later on. Selectivity of peptides versus receptors is fairly low. Let's say that basically dynorphin prefer binding kappa receptors, and enkephalin and beta-endorphin prefer binding mu and delta receptors, but this is not a strict selectivity. It will mainly also depend on the anatomy of the system. So what do these receptors look like? Well, these are G-protein kappa receptors. You heard about that already. These receptors are classically represented like this, a 7 helical bundle floating in the membrane. And well, you heard from Angus yesterday that there's about 1,200 receptors, genes encoding these receptors in rodent. Basically, many of them encode olfactory receptors. There's about 400 genes in the human genome other than olfactory receptor genes, encoding receptors. And 50% of drugs we have in pharmacies encode G protein-coupled receptors. That's important to know. These are primary targets in health research. There's still work to do in the G protein-coupled receptor field because 150 of these genes encode receptors that we call orphan receptors. We don't know their function. We don't know their ligand. So that's a big research field nowadays. So now the cloning of the receptor was in 92, and the structure was solved 20 years later. So we jumped from 20 years to 20 years, right? 73, we had the binding, binding on, ligand binding on receptors. 92, we had the primary sequence of the molecule, and 2012, the crystal structure by the team of Brian Kobilka for the mu and delta receptors, and by the lab of Ray Stevens for the kappa and the ORA receptor. So now we know the structure. Note that the receptors were crystallized, bound to an antagonist. For now, it has not been possible to crystallize an agonist-bound receptor, meaning a receptor that would be under an active conformation. So those conformations that are published today are inactive conformations. And if you think about it, that's pretty obvious. An inactive receptor is supposed to be maybe more stable, less mobile, less dynamic, and easier to crystallize. It is a true challenge to crystallize a receptor under an active form. The binding pocket of those receptors quite differs from binding pockets from muscarinic receptors, for example. It is a broad, a widely open pocket towards the outer extracellular space. And as we said yesterday, there's no allosteric ligand today discovered or known for these receptors. And just to mention this point, I'll come back to this at the end of my talk. The mu receptor crystallized as a dimer. So no one knows whether this reflects any physiological reality, but at least in the crystal, you could see the possibility that those receptors dimerize. And there's an entire literature about opioid receptor homodimerization or heterodimerization, which is well demonstrated in vitro, but we're still looking for an in vivo demonstration. And I'm going to talk about this at the end. So this is the structure. Now, what is the active receptor? And this is going to be the next step in the field. What does an active receptor look like? So there's no way to show you any structure today. Doesn't exist, but I can show you something else. And we need to talk a little bit about signaling complexes, because the concept of a receptor today has extended to the concept of complex or a signaling entity. Just to put things back in a context, for many, many years before, yes?
I just had a quick question on the orphan receptor. Does morphine or heroin bind to that?
To what?
The orphan receptor that you're talking
No,
about?
they don't. The ligand for the ORA receptor actually looks like dynorphin, but does not show the tyrosine at the N-terminus. And a tyrosine at the N-terminus prevents any binding to this ORA receptor. So clearly it's a different set of ligands, and it's a different binding site. This is the view of the pharmacology long time ago, before even before receptor cloning. We knew that different mu agonists, or different delta agonists, or different kappa agonists were able to elicit different responses in vivo. So one couldn't understand this. Why would mu agonist 1 give rise to analgesic response type 1 or type 2? And the idea at that time was that there would be as many receptors as in vivo responses. Today we know it's not true. we only have 3 receptor proteins, a mu, a delta, and a kappa receptor protein. But what we know is that an agonist binding to an opioid receptor will trigger or will recruit G proteins, right, the alpha subunits, GO, GI type subunits, that will in turn engage effectors or may even recruit G protein independent effectors, other effectors, and that these will trigger a number of responses. And you heard about, well, Charlie touched on that a little bit. Activating an opioid receptor will typically inhibit adenylyl cyclase, inhibit voltage-dependent calcium channels, and this will prevent neurotransmitter release at presynaptic site, or activate GERD channels, and this will hyperpolarize cells at postsynaptic sites. So we have short-term effects that, in any case, lead to inhibition of neuronal activity. So you can activate any opioid receptor, you will inhibit the neuron by different means. There's also a long-term effect that will lead to modified transcription. So what we have learned now is that it is not the receptor, the important entity is not the receptor itself. The important entity is the ligand-bound receptor and bound to the signaling molecules. So this complex is the entity that will define the cellular response. And this leads us to the idea that different ligands may lead to different responses. For example, this is a summary, actually we reviewed, Chris and I, a summary of many data, in vitro and vivo data, that show you that a mu opioid receptor bound to morphine will engage a number of downstream proteins that differ from the mu receptor bound to DAMGO, which is an opioid peptide, a mu selective opioid peptide, which engages this type of proteins. And the downstream responses, in particular internalization, for example, will differ. So what is the consequence of this? The consequence is that a same receptor binding 2 different drugs can indeed produce 2 distinct signaling of trafficking responses, and this can lead to 2 distinct behavioral responses. And this is now very much used by the pharmaceutical industry to try and develop what we call biased drugs. That is, a drug that would bind to a receptor but elicit one signaling response rather than another signaling response, possibly to lead to a therapeutic effect or higher efficiency or less adverse effects. And in the context of G protein coupled receptors, so this is very well demonstrated in vitro, in cell lines, this is clear. In vivo, data are sparse, of course. It is much more difficult to show biased agonism in vivo, but for opioid receptors, there's clear evidence that different mu receptor-like drugs can lead to distinct analgesic efficacy or distinct tolerance, less or more tolerance or dependence. In the delta receptor area, and I will show you an example later, we can see that distinct ligand can lead to distinct form of tolerance. And in the kappa receptor field, and Charlie showed you a lot about that, you can see that distinct ligand can produce distinct aversive or analgesic responses or distinct signaling pathway. Okay? So, yeah, I think I will leave the molecular part of the receptors here. Just to basically summarize, today we need to think about a receptor and an agonist and a signaling complex if you want to anticipate and think about a physiological response. Okay, so now let's go to the receptor roles in vivo. So we have 3 receptors that all bind opioid peptides. They all inhibit neuronal activation, so in a way they could do the same things. They do the same things at cellular level. However, what makes their function and they make them different is their distribution in the brain. And each receptor is broadly distributed throughout the nervous system. So to really know which receptor is modulating pain, how do these receptors modulate mood, well, a clear-cut or easy, unambiguous way is to use a knockout approach. So delete the receptor, look at drug responses, and look at animal behavior. So let me just summarize the important thing we have learned from the mu receptor knockout mice. So if we delete this receptor, we can see that morphine is not analgesic anymore. And this is a very, okay, it's an old study now, it's from '96, so that was the first study we made. The idea was to test the wide variety of morphine activities and see which receptor would mediate morphine analgesia, morphine reward, and other effects. This is a simple tail suspension, tail immersion test, and you will see that wild-type mice will withdraw their tails from hot water in 2 seconds, and if you inject morphine 2 and 6 milligrams per kilo, the tail withdrawal latency will increase. So this is morphine analgesia, and this does not happen when mice lack the mu receptor. So the mu receptor is the mediator of morphine analgesia in vivo. And since then, many other drugs that are clinically used have been tested a similar way, and it is clear that all those drugs that are used in the clinic produce their analgesic activity by activating mu receptors. So mu receptors are essential. In a way, without mu receptors, those molecules are inefficient. And if now we look at other activities of morphine, if we look at morphine, for example, conditioned place preference, or morphine self-administration, we'll see nothing happening. Those animals would not self-administer morphine or would not show preference for a morphine-associated compartment. And all other activities of morphine were tested in those animals. The constipatory effects of morphine are absent in those animals. There's no morphine constipation, there's no morphine respiratory depression, there's no morphine immunosuppression. In other words, none of the morphine effects is there when you delete this particular receptor. So the conclusion is pretty clear: a single molecular target for all therapeutic and adverse activities of morphine, which of course drug companies didn't like at that time. This was no hope for the analgesic receptor and addiction receptor, right? It's the same. Everything which is actually used in the clinic today is a mu agonist, and this can be demonstrated genetically that way. Now, we started to think a little bit broader for this receptor function, and thinking about reward processes. We expose these animals to other drugs of abuse that normally do not bind opioid receptors. Okay? So this is just to remind you that morphine conditioned place preference is abolished in those animals. There's no morphine conditioning or reinforcement. If we expose these animals to alcohol, which, as you know and you heard, has many targets in the brain, many targets but opioid receptors, what we see is that alcohol preference or self-administration is strongly reduced. So those animals that lack the mu opioid receptor do not show alcohol reward. If you offer them cannabinoids, and I refer here to a study where THC conditioned place preference was tested. And in this study, it was clearly shown that THC conditioned place preference is reduced, is absent in those animals. They show no THC reinforcement. Remember, THC, the most active ingredient, is binding CB1 receptors. Okay? And now, if you look at nicotine conditioned place preference, you will see that this is down too. This is reduced. So in a way, any drugs of abuse shows, all the drugs of abuse we've been testing show no reinforcing properties in those mice. So if you delete the receptor, you ablate rewarding properties of any drug of abuse. True for cocaine too. Yeah?
Do they have any depression phenotypes?
Yeah, I'm coming to that. So this means that directly for opiates or indirectly, these receptors are essential for drug reward. So the question was, are these receptors also essential for natural rewards? What is the natural, function of those receptors? So what are natural rewarding stimuli? Eating, sexual activity, social interactions. That's the main activities that are essential for the species survival and the individual survival, and that are known as rewarding. You have to learn to do this to survive. If you look at motivation to eat, you will see there's a study about that their motivation to eat is slightly decreased. So it doesn't mean that in a home cage the animal will want to eat, right? Those animals eat normally, but under some conditions they will show less motivation to eat. And there's this study that is very interesting, and I will develop this a little bit later in the second part. This is a study by Francesca D'Amato. She asked for mice a long time ago, and she said, Brigitte, I want to study maternal attachment in your animals. I said, you're crazy, Why would you study maternal attachment? She said, well, social interactions involve reward processes. The mu receptors is important for reward, so I'd like to test whether pups show maternal attachment. Maternal attachment is one of the first early form of social bonding. And this experiment needed about 4 or 5 years. It took a long, long time to produce pups that would be P1 to P8, same age cohorts and so on. But she demonstrated, using several tests, that those mu knockout pups show strongly reduced maternal attachment. And the way you can understand this, or the interpretation is, or was, that so maternal behavior is rewarding, therefore the pup will adapt and then respond, and maternal and bonding will establish that way. So because those pups have no mu receptors, maternal behavior is less rewarding, and as a consequence, the bonding will take place less well.
So
yes?
were these pups born from mu knockout dams?
Yeah.
And so is there differences in maternal care?
No, because she tested that too. It came from the pups, not from the mothers. So basically, these mu receptors are central to reward processes, whether those, whether the stimuli are artificial or natural. You need mu receptors to get a full response on that front.
Can I ask a question?
Yeah.
I know you use knockout, so they are homozygous knockout. When you have a heterozygous, does that response just reduce half, or what happened? Or still complete?
This was not tested. It's too difficult. It would take a long time, so it was not done. Should be done, I agree. I don't know about the gene dosage effect there. So delta. Well, if you repeat similar experiment with delta knockout mice, you see very different responses. Those mice are totally different. And you will see that delta receptors are not essential for reward. If you look at morphine self-administration, you will see that those animals self-administer morphine as well as control animals. So there's no deficit in morphine reward or motivation for morphine. If you look at alcohol intake, you will see that those animals actually drink more alcohol, just the opposite of mu receptors. If you look at THC condition place preference, no phenotype is detectable. if you look at nicotine self-administration here, you can see that there's a slight decrease. So this pattern of responses is very different, and it is clear that delta receptors are not central to the reward process, but modulate those processes in a way we don't understand. Now let's go to the emotional responses, depressive-like state or anxiety. So we talked about those models. We know the models are not great, but have predictive value, okay? If we look at the 3 knockout lines and we compare responses of these lines, so I just show you the prototypal test, the dark light box and the forced swim, only delta knockout mice show a strong phenotype. The 2 other lines don't show much. And what you will see is that delta knockout mice show strongly increased latency to enter the lead compartment, which means higher level of anxiety. And you can see this in many other tests. I only show you one test, right? There was a whole panel of tests. And this is also detectable on several genetic backgrounds. It's a very strong phenotype. And in the forced swim, same thing. You will see increased immobility, which means a despair-like behavior. And you will not see this in kappa knockout mice, which is not surprising if you remember what you learned from Charlie Shafkin's talk. The kappa dynorphin tone gets up, or the system gets tonic, the kappa system, only under stressful conditions. Under home cage conditions, the kappa system is pretty silent, so we see no phenotype. And the mu receptor knockout might show no, if not a slightly opposing phenotype. So this is typical for delta receptor activity. And from there, so this to summarize, we have a complex regulation of reward processes by delta receptors, and we clearly have a tonic, anxiolytic, and antidepressant activity. And from that set of results, delta agonists that were developed in drug companies to treat chronic pain were sent to the psychiatric departments, and there are now delta agonists under clinical trial to treat depression. So the delta receptor has an entirely different activity profile compared to the mu receptor. Over in the brain, those 2 receptors bind on enkephalines, right? But they're very different in their function. So this basically summarizes what I told you. I didn't show you anything about kappa receptors. You heard most of the the most relevant, significant work by Charlie Shafkin yesterday or 2 days ago. If we want to think about these receptors, both the genetic approaches from us and others and the pharmacological approaches that confirm, they all confirm each other, tell us that along a reward, I would say reward dysphoria scale, clearly high mu receptor activity means high reward, high kappa receptor activity means dysphoria. And if we now look at a mood scale, you can think of delta receptors or high delta receptor activity as being anxiolytic or antidepressant, while kappa receptor activity is just the opposite. Now, how do you fit this in the addiction history? So this is just to summarize what you heard before by many speakers, and this is the cube circle. And so, of course, you know that, well, recreational or social use is not addiction, but is rewarding, but does not mean there is any addiction-related behavior. For some individuals, this may lead to binge intoxication, enter the addiction cycle with this acute withdrawal aversive state, preoccupation craving, back to binge drinking or consumption. And some individuals are able eventually to maintain protracted abstinence, and we know that in the long term this particular state is characterized in humans by lower mood, high irritability, and hence emotional responses, and that this indeed is the clinical problem how do we set opioid receptor activities there? Well, how do they contribute to neuroadaptation in the system along the development of a history of drug abuse? Clearly, mu receptor will contribute to recreational drug use and potentially to the initiation of addiction or addictive behavior. Clearly, the kappa receptor and dynorphins, the kappa receptor tone will increase and will contribute to a relapse. And maybe, but this is not demonstrated, maybe delta receptor activity, which is anxiolytic and antidepressant, counteract some aversive states of protracted abstinence, to be demonstrated. This is speculation. Okay, and to finish with this part, we wonder whether these receptors may be important for other aspects of addiction. That's just another way to look at circuitry, addiction circuitry. You know that, well, you know all these areas, and basically modifications throughout the circuit will contribute to addiction, such as lower reward processes. You heard a lot by George Koob about that. Mood is lowered, stress responses are enhanced. You heard about other speakers about that. Motivation is entirely directed towards drug use, so this goes here. And then habitual behaviors are strengthened, and David will tell you a lot about these things later on. And also you heard about inhibitory control being weakened. And this occurs in cortical area, and if you look at opioid receptor expression, so these are sagittal sections, and basically what you see here is radioactive ligand binding to sections, to brain sections. So kappa, weak binding, mu, this is typically mu receptor binding on a rat or a mouse brain. This is a mouse brain, and you see that there's strong expression here and in the cortex. And this is delta receptor expression seen by ligand binding. And you see that in both cases, there's a strong expression in this prefrontal cortex. So this led us to ask the question whether this delta and mu receptor may be involved in inhibitory control or impulsivity. And I'd just like to mention this little study that was done by Celluarl, who came for a sabbatical in our lab. And she was used to deal with humans and rats. And she came to me and said, I want to see what I can do with mice. And she tried to do the hardest thing you can do with mice, which is what I want to show it to you, is to study motor impulsivity. You know that mice are extremely active. So trying to teach mice to refrain from nose poking is a true challenge. But she was able to do it. So basically the task is the following. The animal is in an operant chamber. The animal will learn to nose poke. Well, we first learn where is the so it's not sugar pieces, right? This is liquid sucrose that is presented to the animal. The animal will train to work for, this is a FR1 schedule, to nose poke once to get one reward during 40 minutes, and then the animal will repeat this every day until he succeeds 25 trials, and then we'll make it a little bit more difficult, FR3, 3 nose pokes to get one reward, and then in phase 3 we will teach the animal that the reward is not available all the time, it is only available 5 seconds every 30 seconds. And in this training period, the animal, well, inappropriate nose poking will have no consequence, but the reward will only be available 5 seconds every 30 seconds. So this is a training period which lasts like about 2-3 weeks. The animal will learn to go through all this, and we trained mu knockout mice, delta knockout mice, and we trained cohorts on 2 different genetic backgrounds, a hybrid background or a black 6 background, and all the animals progressed at the same rate. Okay, under those conditions.
How long does it take?
About 2 weeks.
Only 2 weeks.
Yeah? Well, she was pretty good. We try. Other people tried later on, didn't work as well. So you need to be a little bit skilled.
Is that
7 days a week? I heard they learn faster if you do it every day.
Well, yeah, each step is about between 2 and 5 days. So the problem is that the animals, individuals don't progress at the same rate. So in the end, you end up with each individual at a certain stage of the training. So it's getting pretty complicated. But basically now all the animals have trained. So now they will go through the test. And the test is the following. Nothing happens for 20 seconds. Then there's this, you don't see where, pre-Q 1 to 8 seconds. Then nothing happens. There's a variable time. Remember, the animal is used to know that something will happen now. But the light doesn't come up. And during this variable time, should the animal nose poke, error. It goes back to the beginning. The animal should not nose poke until the lights come up. And then for 5 seconds, the animal is able to, the animal nose pokes and gets a reward. If the animal does not nose poke here, again, it goes back to the beginning of the session, but they always nose poke when they see the light. And then they get their reward. So basically what you measure, so what I'll show you is the data every day. So it's a 40-minute session. And the parameter I'm going to show you is the efficiency ratio, which means the number of reward, the ratio between number of rewards and number of nose poke. So if you're really bad, means 0. 0 means you got no reward, means you nose poke the wrong time each time. 1 is the best score because each nose poke produces a reward. Okay, so you will be between 0 and 1. And typically mice, can go up to 0.4, right, in the literature. And what Celluarl was able to do, so these are the mu-knockout mice, and these are the wild type. So you see every day they learn, and the wild type here get a score of 0.5, which is really good, actually. So the test works well. And what you will see is that the mu-knockout mice do better, significantly better. They work like machines, they wait, light comes, they nose poke, no problem. Very good self-control. So these mice have no mu receptor, right? And the delta knockout mice are terrible. Look at the score. They nose poke all the time, they nose poke inappropriately, and they gain very few rewards. This is very new because this shows, first it shows that the two receptors have very different function, and in this case, opposite functions. And then it takes us into another, basically, areas of mental health disease or brain disease. We come up to impulse disorders area. So to summarize, high mu receptor activity means high reward, but may mean also high impulsivity. High delta receptor activity means low anxiety and low impulsive behavior. So we now have these receptors in 3 areas of psychiatric disorders. So that's pretty new.
It's a little counterintuitive, right, that
Yeah.
the mu receptor knockouts
Yeah.
perform better. Because you would think they would have diminished reward signaling or diminished reinforcement. So this is really cool. And so we think about
I think that the function of the physiological function of the mu receptor is much more complex than what we think. And heroin or morphine are such strong drugs, and the rewarding effect or euphoric effect is so strong that it basically overcomes everything. So when you go into the physiology of this receptor, you discover, and actually this makes sense if you look at the receptor distribution, that it regulates many different processes.
Do you think it's worth trying to repeat these studies with the peptide knockouts to try and further refine where this could be happening, or would you just go to the conditional?
No, I would rather go to the condition. The peptide knockouts are, this is a little bit difficult. There are knockouts for proenkephalin, for prodynorphin, for beta-endorphin, but the peptide phenotypes do not match the receptor phenotypes for 2 reasons. One reason is these mice have never been tested by the same experimenter under the same laboratory setting. So if you really want to draw a conclusion, you should take all these mice and run them through the same test. The second thing is the peptides probably act at several receptors. So difficult to tease out which is doing what.
I have another question. It seems like your immune knockout is more smarter, like intelligence.
Yeah.
Those so-called performance drug, I wonder if this really works to enhance the performance.
No, well, you're talking about memory performance or learning. Well, they do not better in the training here. And in the plus maze, this is a study, in a water Morris maze, which is a study we did a long time ago, they don't perform very well. So I don't think it's a general, they are not smarter, I don't think so. Well, the next 10 minutes, I'm just going to move here. So, yeah, where do these receptors operate in neuron, in all those circuitries? So I have just 2-3 slides. Of course, the next step is to go to conditional knockouts. For these receptors, we've been starting with looking at pain. So this is not addiction, this is a pain study, but very briefly. We know by looking at pain phenotypes of these animals that delta knockout mice show increased pain responses under condition of persistent pain. So this is a receptor that comes into play when pain is chronic and is working to reduce pain under chronic pain conditions. Very different from your receptors again. So pain, as you know, is conveyed from the periphery. The noxious stimuli are conveyed from the peripheral tissue, skin, or others, through these, what we call the primary nociceptive neurons, here with cell bodies at the level of DRGs, and then this signal goes to the spinal cord, and then brainstem, thalamus, cortex, where pain becomes conscious. So the opioid receptors are expressed all along these pathways. So one simple question we asked was, well, what is the contribution of peripheral receptors versus central receptors to analgesia, to opioid analgesia? And when I'm talking about peripheral, the primary nociceptors I'm talking about here with a cell body in the DRG is in the peripheral nervous system, right? And then projects to the spinal cord that is part of the central nervous system. So what we did is we used Cre mice that express Cre in small nociceptors in dorsal root ganglia, and we deleted mu and delta receptors, specifically in that neuronal population. So I'm just going to show you the data with delta, because these are more interesting. Basically, the conditional knockout shows, we don't see much here, right? The conditional knockouts show strongly reduced expression of the delta receptor, specifically in small cells of DRGs, which are the nociceptive neurons. And if we expose these animals or study those animals in a model of chronic pain, this is a model of inflammatory pain. You inject fluid adjuvant, and then the paw will get inflamed, and you will measure pain responses or pain sensitivity. You can see that by poking the paw, the mechanical threshold day by day will decrease. Means the animal will be more and more sensitive to a mechanical stimulus. And this is the white square here, is the increased pain response in control mice. And you will see that mice that have no delta receptors, specifically in small neurons of DRG, show enhanced pain response. Pain is stronger. So this means this receptor normally tonically inhibits the pain in that particular neuronal population. And you can see a similar thing in neuropathic pain. And then we tested the analgesic effect of a delta agonist. You inject a delta agonist under those conditions, you will reduce pain. And so this is in the model of inflammatory pain. This is a normal pain threshold. These are animals that have received CFA. so the pain threshold is much lower. Here is the effect of SNCAT. The analgesia is, that's the analgesic effect, dose-dependent effect. And what you see here in those conditional knockout animals, SNCAT is not analgesic. There's no analgesic effect at all. And this is true both in the inflammatory pain model and in the neuropathic pain model.
So baseline nociception isn't different?
No. So basically what this tells you is that this small population of delta receptors in this particular neuronal population is by itself is able to reduce chronic pain and mediate the entire analgesic effect of the delta agonist.
What are you using for the neuropathic pain, nerve ligation?
Sciatic nerve ligation, yeah. And this is ongoing. So we have been trying to delete by conditional knockout receptors in the brain. So we have been targeting what we GABAergic neurons of the forebrain. We know opioid receptors should be expressed mainly in GABAergic neurons. And what you see here are ligand autoradiograms of mu receptor binding in those conditional knockouts. So what you see down here is the control animal, and you see the strong mu receptor expression in the striatum and at the level of thalamus and habenula. And if you look now in the conditional knockout, receptors have disappeared in the striatum, but are still there in the thalamus, in the habenula, and are still there in the brainstem and the spinal cord. For the delta conditional knockouts, so this is reversed. Actually, the control mice are on the top. This is a classical delta receptor expression or binding, a ligand binding pattern. And what you see here is the normally strong expression in the striatum and the basolateral amygdala. And here, in the conditional knockout, receptors are gone, totally gone in the olfactory bulb, totally gone in the striatum, still intact in the basolateral amygdala. So now we can play with these mice and try to ask what are striatal receptors, what is their function. And just first data show that clearly the locomotor effects of heroin are gone in those animals. Normally heroin puts the animals to run. They don't run anymore, but morphine analgesia is perfectly intact. And here, and this is Paul's work, he may tell us a little bit about that later on, we can see that the hyperlocomotor activity of SNCAT, the delta agonist, is totally gone. But the emotional response, yeah, you can tell about that. So the other reason why I show you this is, look at this. This is miserable. How can you see a receptor and see where a receptor operates if you do a ligand autoradiography? That's the only way we have today to quantify receptor expression. There's no antibodies, or there's very little antibodies we can use to actually see the receptors. Which cells express these receptors? Well, we don't know. The only thing we can say here is that they are gone in GABAergic neurons. Okay, so they should have been in GABAergic neurons. But the resolution of ligand autoradiography is very low. So how can we see where the receptors are expressed? Which neurons, where on the neurons? And this leads me to this tool. Maybe we can close a little bit the curtains, because otherwise you won't see anything. Oops. Yeah.
The other one's still?
Yeah, could you close this a little bit? Yeah. So, what you used to see are reporter animals that have fluorescent cells, right? You've seen this in many previous talks, and classically what is being done in neuroscience, I think that's okay, that's okay. Or, well, green mice or red mice or whatever you call those mice, typically are reporters for cells. What we try to do is very different. We try to do mice that will report for the receptor. So what Greg Scherer in my lab has wanted to develop, and that's, well, a good example of a student that really wants to do a high-risk project, and I told him, no way, you don't do this, it's never going to work. And he insisted like crazy. And he was right, really, he was right. So Greg said, okay, why don't we fuse a GFP to the C-terminus of this receptor and create a knock-in animal where we would replace the receptor, the native receptor, by a fluorescent receptor. And before we did that, he verified in cell lines that the function of the receptor is retained. So this receptor is able to bind ligands, to signal, do everything normally, at least in cell lines. So we embarked on the knock-in line. I will not go into detail. This is just a classical homologous recombination approach. Just took 4 years. That's just 4 years. And of course, after 4 years, we get the first animal who say, well, is the receptor expressed? Because you might, when you modify the genome, you never know what's going to happen. And typically, when you go for a knock-in, you think you've done everything right, and you end up with a knockout, because the gene doesn't like the modification, and somewhere transcription factors have decided that this gene should be silenced, this gene, right? So we found mRNA, great, mRNA is produced. We found the protein, and there was no, and we could only detect the fusion protein. So now actually, we don't need a delta receptor antibody. We can use a GFP antibody to detect the receptor. And the binding sites were normal. Okay, so we have an animal which expresses a receptor at physiological levels. And what do we see? Well, we can do a lot of things. We can do anatomy, we can do dynamics, we can do physiology. So I will quickly go through this. Well, if you look at the whole brain, that's what we see. Okay, a fluorescent brain, good. Well, it was amazing because remember that this is a knock-in, so it's a single copy gene, so the expression level is very low. So I personally didn't expect to see anything. So I sent Greg, kill another mouse, you know. He came back. We saw this again, saying, well, incredible, you know, but we can see this signal. So if you go for brain section, what you will see looks like what you see when you do ligand autoradiography, right? Strong fluorescence in the cortex, in the striatum, in the BLA. But now what is interesting is we can slice those, you know, take those sections to the confocal, and now we can start seeing individual neurons. So what you see is not the neuron, is the receptor labeled in the neuron, right? So the patterns are very different from what you would see from a labeled cell. And then you can zoom in, and then you can label with GABAergic, cholinergic, whatever marker, and see, for example, here in the hippocampus, that this neuron is a GABAergic neuron, and you see on the surface, this green fluorescence is the receptor on the surface. So now we can revisit, or basically study the receptor expression throughout the brain, and spinal cord, and DRG, and nervous system. And actually this was very important because Greg used that, for example, it was important in pain research to show that the delta receptor does not colocalize with substance P, for example. So I won't give you the whole background, but there was a whole theory that the two substance P and delta receptors were co-expressed. But Greg looked in, you know, these mice and saw that there was no colocalization. So he made a lot of enemies with that. But you know, this is a way to revise a number of theories. Now you can see, for example, those receptors in the GI. You know, no one had never seen this before. There's a lot of this receptor here, and you can, for example, label neurons for pre- and postsynaptic compartments, and this is a postsynaptic receptor because it colocalizes with MAP2, and this is an axon because it does not colocalize with MAP2, and so on. So just to show you example. But now you can go and fine map the receptor in neurons. Now you can also take neurons out of the brain of those animals, do primary culture, and then you have single layer culture, and you will see those receptors nicely concentrated on the surface. There's a lot of green color inside because, of course, these receptors are produced, synthesized, green. Okay. So what you see here are receptors being synthesized on their way to the surface or on the way back inside. you can phenotype these neurons, know whether you have a GABA neuron or a cholinergic neuron or whatever. And what is even more interesting is you can look at this receptor trafficking, because the receptor is naturally fluorescent. So this is a neuron from a striatal neuron, and this, so it's a little movie, and so you see the receptor is here on the surface. Now the cell is exposed to delta agonist, and what you see is the fluorescent clusters and enters the cell. So that's the receptor trafficking, and after 20 minutes, there's nothing visible on the surface anymore. It's all inside. So this is normally 20 minutes, right? You got a kind of accelerated movie.
So does it recover?
Good question. Takes a while, and I come back to this. In vivo, it takes about 24 hours. So
Like
this,
which takes like 2 weeks.
so you'll see. So it goes to lysosomes. You can label, and this is a lysosomal labeling. So it looks pretty uneconomical, right? You activate the receptor once, it goes to the bin, basically, right? Goes inside, trashed. We start now developing this time-lapse, real-time experiment in slices. It's technically difficult, but we're getting there. So what is the consequence exactly of this in vivo? What does it mean? Does it have any consequence on behavior? And I'll summarize briefly Amina's work. What can we do? So you cannot see real-time receptor trafficking in a live animal. We've been trying to put an optic fiber, but today the confocal microscopy, ambulant or confocal microscopy is not resolutive enough for that type of signal. So we just see green. So what we have to do, what we can do is we can treat an animal with a drug, measure behavior, analgesia, anxiolytic response, and then immediately after kill the animal and look at the receptors throughout the nervous system. So typically SNC 80 treated animal, you will see these clusters. So you will see these cell bodies throughout the nervous system with highly fluorescent vesicles, while in cell line injected animal you will see clearly see, you're not used to it, but clearly see the fluorescence on the surface. And you can quantify the internalization from there. And what Amina used are two different ligands. One is the SNC80 I told you about. This is the prototypal delta agonist. Everyone is using the commercial ligand. And here, this is another one we got from AstraZeneca, which is extremely interesting because it has the same affinity for the receptor, same efficacy and potency to activate G proteins, same analgesic efficacy, same anxiolytic efficacy. So everything is the same, except it does not internalize the receptor in vivo. So, summarize in one slide, what happens? If you inject the SNC80, the high internalizing receptor, you will produce analgesia, you will produce anxiolysis, okay, depending on the test you're using. And if you look at the receptor, after that injection, it will be internalized and committed towards degradation. If you try not to inject the drug again, no effect at all. The animal is totally tolerant. And if you wait 24 hours, the receptor store has restored, so the cell has produced receptors. But if you now repeatedly do this every day for a week, the cells cannot catch up. Too much degradation, not enough recovery. And basically, where you get to is an animal that has no more receptors. And if you look at the fluorescence, you don't see it anymore. And of course, this animal is generally tolerant. Now you can inject whatever you want and test anything you want, nothing will happen. This is a classical view of tolerance. Now, this one is very interesting. This compound does not internalize. So you will inject the compound, you will get the behavior response, no problem. And you will see after the injection that in every neuron throughout the brain, the receptor stays on the surface. It's there. And then you can inject again, you get the response, the receptor stays on the surface. So you would say there will be no tolerance. So after chronic treatment, after a week, we look at the receptor, still there on the surface throughout the nervous system. But we see tolerance to analgesia. On the other hand, responses, anxiolytic responses, are totally intact. What has happened is that in a long nociceptive pathway, and we've been able to show that in DRGs actually, the receptor is still on the surface, but the effector has uncoupled. So voltage-dependent calcium channels basically are desensitized. They don't couple to the receptor anymore. So the neuron has adapted by uncoupling the effector. But this does not occur in the brain, and the anxiolytic response remains intact. So here you have a way, by playing on internalization, to produce a drug or to develop a drug that will be efficient for anxiolytic purposes without tolerance, for example. Okay, so that's one implication. And I think, this is, well, that's an important message. I will stop after that. This is all drug treatment. So if you inject a drug like SNC80, you will see the receptor internalizing throughout the nervous system and be degraded. This is a pretty tough treatment. Does this happen when neurons release enkephalins? What happens under a physiological situation? So it took us a long time to actually find a behavioral situation where we would see receptor internalization under endogenous peptide release. We thought it would be easy. We started, we produced pain, we pinched the animals, we produced all kind of pain. We looked in the spinal cord, we saw nothing, no internalization. Then we threw the animal in force swim, stressed the animals, we saw no internalization. So it took us a while to find an appropriate situation, and this was what we call context-induced withdrawal. So we made the animal dependent on morphine, and then put them in a context where they received morphine without morphine, and they would show withdrawal signs induced by the context. And we knew from the literature that this produces endogenous peptide release in the hippocampus. Say, okay, let's do that. And we did that, and we found that in very few neurons, so 9% of the neurons of the CA1, very region-specific, we could see some patterns of internalization very transiently, from minute 30 to 45. So it is a very subtle phenomenon. And in addition, it is very different. It looks like this, not like this. So you may not see the difference, but what I can tell you is that you don't see vesicles, you see a fuzzy pattern. So the internalization compartment seems different from the internalization compartment we get when we use a drug. So what this tells you is that what a drug does is very different from what the endogenous compound or ligand does on receptor trafficking. That's important. This is, it's like in drug abuse, what a heroin will do on your reward system is very different from what food will do, on your reward system. And this is a kind of cellular demonstration of that.
Were those restricted to specific hippocampal layers? The
Yeah.
cells that responded? And that
Yeah.
Presumably they're GABAergic interneurons?
Yeah.
And
Yeah.
do you know which layer they're in?
This was in CA1 and CA3, but I don't remember, I can't tell you. So now what we're trying to do is using slices and then stimulate neurons, afferent neurons, to trigger enkephalin release and monitor in real time the receptor internalization. Very tricky.
That's hard.
That's very tricky.
Because I think the enkephalin's all in one particular region of the slice coming in from the cortex.
Yeah.
But there are a few interneurons that can release it too, so I don't really know which supply.
We think this is what we call the temporoharmonic connection.
Exactly.
That's where the enkephalin comes from.
Right.
And that's
like
where,
very
that's
far
what we try, yeah.
from a lot of the cell bodies, so it's an
Absolutely.
interesting case for volume
Mm-hmm.
transmission.
Yeah, absolutely.
Well, when you do those experiments, do you need to use the GFP antibody, or can you see the fluorescence well enough without?
We see it better with the GFP antibody. So all experiments that we do on fixed tissues, we would use an antibody to make sure. But you could see it. Well, it depends on the performance of your microscopes. It's not a huge signal. So we have been shipping those mice to, several people and sharing the mice. And people think they will see, bright green fluorescence like any transgenic EGFP mouse. No, you have to. It's a subtle signal. So maybe we can make a break here. Yeah.
Just one more question, because in the very beginning you also show morphine and dango have different response and
Yeah.
of a couple resistant. So when you show that the last slide, you treat morphine and CA1 and CA3, you see only 9% of cell have internalization.
Yeah, this is not
How about
morphine.
the dango? Do you have like
No.
global?
This is a delta receptor. It's not a mu receptor. So morphine and dango are compounds for the mu receptor. This is a delta receptor. So we have two other types of compound, the SNCAT and the RM. I showed you internalizing, non-internalizing. So it's a similar situation.
So morphine and dango also has different internalization pattern. It can be found in the hippocampus. I don't know if you have that mouse.
Well, we need the animal with a fluorescent mu receptor. And I'll show this after the break.